Okay, well, hello everyone. It is really great being here today as part of this interesting event or getting a slot in 24 hours of life science. My name is Jana Ognjenovic and I work at the Frederick National Lab for Cancer Research as part of the National Cryo program. Today, I'll be talking about our experience in getting to high resolution using Cryo. One of our missions is to investigate novel and less explored emerging cryo technologies, and one such example includes our work on exploring cold PEG technology available on Joel's cryo microscope. We want to compare how it compares to the standard Schottky PEG for high resolution cryo data collection. The Cryo 200 microscope is equipped with an in-column omega energy filter to remove inelastically scattered electrons that would otherwise just add noise to the image. And our microscope has Gatan's K3 camera installed, which was chosen because of its high frame rate that offered the potential for maximal throughput when using counting mode. And the goal here was to use this combination of hardware highlighted by the cold PEG to produce structures that would have density displaying some ultra high resolution features, such as those shown on the slide here around the picture of our team. At the time when we started this project, there was a lot of skepticism around cold PEG, as despite the possible theoretical advantages, there were no practical demonstrations of its usefulness. And a lot of people actually thought that cold PEG data collection with the Cryo was more trouble than it was worth. But we wanted to explore potential benefits of collecting Cryo data using a source with such high coherence, especially once it became obvious that automated tip flashing with the software would actually keep the beam intensity sufficiently stable over the course of data collection. And great news on this technology actually came online in the form of a tweet posted by Takayuki Kato, where he reported a 1.76 angstrom resolution of apoferritin done on their Cryo 300 microscope using K2 camera. And this was only done with 56 images. Later on, this result was updated when the rest of data was added in, totaling 840 images from a single day of data collection. And this led to 1.54 angstrom resolution of apoferritin. This was the world record for the highest resolution single particle cryo map at the time, and provided a strong evidence that the smaller energy spread that is offered by cold PEG produced real benefits in the ultra high resolution range. A bit later, Dimitri Teguno processed the same data set using his program M with EWOLD aware CTF refinement and got apoferritin to 1.34 angstrom resolution. This result was published this year. Despite this result, there were still questions about the feasibility of getting Sub-2 angstrom resolution with a Cryo on a lower symmetry particle. And there were also concerns about changes in beam tilt upon adjustments in objective lens setting and or tip flashing. Still, this apoferritin result made us even more excited to get the microscope installed and to see what resolution could be achieved on a more technically challenging sample. So we decided to use BetaGal as our initial test specimen, since this sample has lower symmetry than apoferritin. And the main change we made to the data collection scheme compared to the standard data collection procedures was to simply use the stage height for focusing instead of the objective lens. This way, the setting of the objective lens was left unchanged during the data collection, so we did not have to worry about this affecting the beam tilt over the course of the session. And there were obviously a lot of concerns about using the stage instead of the lens for focusing, mainly related to its effects on accuracy, throughput, and stability. Luckily, we had Takuma Fukumura stationed here at FNL with us. He's a Joel's applications engineer, and he was able to write scripts for CRLEM data collection to deal with most of these potential issues. And once we analyzed the data, it was apparent that the data quality was largely unaffected by the change in focusing method. Importantly, we were still able to use normal image shifting to collect multiple images for each stage position, which allowed us to collect about 5,000 images per day, enough to produce a 1.8 angstrom BetaGal map from a single day of data collection. And we published our findings last year in IUCRJ. And here are just a couple of regions from our 1.8 angstrom BetaGal map. As usual for maps in this resolution range, you can start seeing some high resolution features such as holes in rings, nicely defined carbonyls, ordered water molecules. Here you can see a nice interconnected water chain that sits at the interface of these three regions of protein. Overall, we were able to assign over 3,600 water densities in the map. But if you look closely, in some instances, it seems like there is some protrusion, some bumps coming from the density bumps. And this was not a noise because we know that this map actually has really low level of noise. And actually those bumps in density, they coincided with the location of hydrogen atoms. And while we were not able to resolve individual hydrogen atoms or even larger atoms, it was still really informative to use these density bumps in downstream steps. And I'll show you in the next slide what I mean by that. Here you can see a practical upshot of getting a map to high resolution. The panel here shows examples that illustrate the importance of being able to visualize the density for the hydrogens and the side chains of glutamines, asparagines, and threonines. This is the well-known Enqueue problem in structural biology. It's concerning the ambiguity of placing the fitting of the amide group as the amide nitrogen and oxygen atoms. They have similar densities and they are indistinguishable in the maps at moderate and low resolutions. And it is estimated that the average error rate of fitting the Enqueue rotamers is around 20%. And there are even some software packages like Enqueue Flip that are dealing with this kind of issue. And they're able to flip any incorrectly assigned Enqueue rotamers. The BetaGal map we obtained using Cryo 200 microscope shows that in a 1.8 angstrom Cryo map, you can often easily distinguish between incorrect and correct rotamers merely by visual inspection due to the presence of the additional density coming from the hydrogens. When we compared our Enqueue orientations to those obtained by X-ray crystallography at 1.6 and 1.7 angstrom resolution, we found that there were several rotamers that had been flipped. And there were even on several Enqueue side chains where the orientation actually differed between 1.6 and 1.7 angstrom X-ray structures, demonstrating that the ambiguity persists even into the high resolution range of X-ray structures, and that although our BetaGal map is nominally lower resolution, in some ways it contained a bit more structural information and ultimately led to a better model. And to give you a better idea of the types of features we are seeing in our 1.8 angstrom BetaGal map, here is a panel showing an example sidechain density for each of the 20 standard amino acids. And as you could see on the previous slide for glutamines, asparagines, and threonines, those density bumps corresponding to the location of hydrogen atoms, they are visible on all other sidechains, from small ones such as glycine all the way to big ones such as arginine, tryptophan, and even the acidic ones show nicely resolved density for corresponding to location of hydrogen atoms and for carboxyl as well, even though they are known to be the most prone to radiation damage. And this is the image of Taku and Alan setting up data collection on our Cryo 200 microscope. So far, we have been collecting all of our data using Serial, and Taku actually wrote all those scripts, and they are freely available to anyone who would like to have them. We are currently working with Gatan to expand the data collection software to include Latitude as well, which would allow more freedom when it comes to data to choosing data collection software. So as I said, all of our data that I'm going to show you today are collected with Serial EM. And JADAS, Joel's proprietary data collection software, was used to collect 1.34 angstrom alpha-ferritin data. So we know that data collection software can be used for high resolution data collection. Although for that data set, the collection was done using only stage shift. So some of the common questions we are getting about Cryo 200 microscopes are basically, okay, so you publish 1.8 angstrom BetaGal map. Does that mean that this is the resolution limit for Cryo 200 microscope? And we are still not close to 1.34 angstroms done on alpha-ferritin on Cryo 300 microscope. So this is something to shoot for. Even just looking at 200 kV structures, the record for the highest resolution at 200 kV is still the 1.7 angstrom structure of alpha-ferritin done on Glacius. And our 1.8 angstrom structure of BetaGal is very close. However, later on, I'll talk more about our efforts to improve the resolution there. Another important question we are getting is if it is possible to collect higher quality data with a tilted stage on the cryo arm, whether it is tomography tilt series data or even just tilted single particle data, which of course is sometimes needed to overcome a preferred orientation problem. And David Bell actually has shown some nice tilt series collected on their cryo arm 300 microscope in Glasgow. So we are looking forward to collect some of tomography data on our cryo arm 200 microscope as well. Taku wrote all the scripts, and we hope to do this soon. And another question we are getting is that, okay, showing that the high resolution structure can be obtained for BetaGal was a nice technical achievement, but can the same be done for some biologically relevant sample? And fair enough, structure of BetaGal has been done at high resolution before, both by cryo-EM and crystallography. But this was a necessary first step in developing method to then afterwards tackle some biologically relevant samples. And one such an example of, I would say, biologically extremely relevant sample is a human ribosome. And it's especially timely right now where I hope that all of you who got their mRNA vaccines, their ribosomes are happily producing enough spike proteins to keep you happy. So for this experiment, we actually purified human ribosome, unstalled human ribosome directly from HeLa cells. And here's a representative micrograph where you can see recognizable ADS particles. And those strings surrounding particles, they are actually mRNAs that were caught inside the ribosome at the moment of plunge freezing during the grid preparation procedure. The ribosome sample is known for its heterogeneity, especially the way it was purified for this experiment. That's not, it was unstalled. So during data processing, we had to employ multi-body refinement in Relion to account for relative flexibility between its small subunit head and body domains and a large subunit domain. And here just the mask showing how which masks to use for multi-body refinement. And this actually led to 2.1 angstrom resolution for the ADS particle. When you look at this local resolution plot, you can see that, yeah, 60S particle, we were able to, resolution was just below 2 angstroms, so sub 2 angstrom. And if you look at the local resolution plot, you can see that in the core of the particle, the resolution extends actually to 1.8 angstrom resolution. And as we could see for our 1.8 angstrom BetaGal map, at this resolution, you can start seeing a high level of structural data, nice features, including density bumps corresponding to position of hydrogen atoms. And here on the right, you can see that we are actually starting to see really nicely ordered solvent structure. Nicely resolved density allows more accurate placement of waters and metal ions. Also, one could start inferring metal ion identities based on interatomic distances and coordination geometry. And here's an example of hexa-coordinated magnesium ion, where ligands are arranged in a recognizable octahedron. Next to it is a hexahydrated magnesium ion. And using map of this quality, we can begin placing in highly coordinated potassium ions, which are distinguished by a coordination number and well-defined interatomic distances. The placement of zinc ions is usually trivial. And here you can see an example where zinc ion is coordinated by 4 cysteine side chains. To give you a more complete picture of the quality of the ribosome map we obtained, here is a full amino acid panel. And similarly to what we showed for the 1.8 angstrom BetaGal map, here you can see that although the overall resolution of the map is still not as high as it was for BetaGal, in the best parts of the map, we are starting again to see bumps corresponding to the location of hydrogen atoms, along with the usual high resolution features, such as holes in the rings and nicely defined carbonyls. And just as for the BetaGal map, the presence of the hydrogen density in the new map help us to more easily identify the correct rotamers for glutamines and asparagens. And similarly, the bulkier density from the 3 gamma carbon hydrogens of the threonine makes fitting of this side chain pretty straightforward. And given that the sample is ribosome, next to the amino acid panel, here is additional panel showing the density around 4 standard nucleobases. And moreover, at this resolution, visualization of post-transcriptional modifications it becomes apparent. There are many PTMs in human ribosome, and here is just a panel showing 13 distinct types of PTMs present in human in the map we are seeing. Nucleobases, they play an important structural and functional role in ribosome, and in the best parts of the map, we can start seeing those, as I mentioned, density corresponding to location of hydrogen atoms, and we can also start visualizing interactions between nucleobases in great detail, as shown here, a canonical Watson-Crick base pairing, as well as a non-canonical base pairing. So it has been known for a long time that there is a backbone modification in US11 protein in a small ribosomal subunit, but it was only seen for the first time last year in a paper published Watson last year from Jamie Kate's lab. They reported to Angstrom resolution of E. coli ribosome, and in this structure they could clearly pinpoint where the location of this isoaspartate. In the same paper, the authors speculate that this isoaspartate actually is being universally conserved. And indeed, in our map, we could actually see that fitting of isoaspartate in the same protein looks really nice. So that's one example of backbone modification. And another example here is post-translational modification of histidine, methylation of histidine. It's interesting, this example is pretty interesting because this methylation, we tried to confirm it by using mass spec, but it was like no matter which enzymes we used to digest the peptide, the mass spec just would not pick it up. But this methylation of histidine is actually known in yeast ribosome. So I guess another example showing why getting a high resolution is actually pretty important. So this is another example of another project we did on ribosome. As I mentioned, ribosome has a really important role, especially today. So this is an example showing what happens with the ribosome when the SARS-CoV virus enters a cell. One of the ways the virus is hijacking the host cell is through the protein that is among the first proteins to be made out of viral genome. It's non-structural protein 1, NSP1. This protein is able to inhibit host gene expression by inserting its C-terminal domain around 30 amino acids into the mRNA channel of the ribosome, and basically prevents mRNA from accessing the ribosome, thereby reducing protein synthesis. In fact, the structure of this complex has already been done and published. It's been done by 4 different groups, all on 300 keV microscopes, and all in 2.5 to 3 angstrom resolution range. Now, in our new structure, the resolution is already higher, at about 2.2 angstrom resolution right now, but this is still work in progress. Clearly, this is still in early stage, so we definitely hope to push the resolution further. But even here, at this point, we can start seeing some new features that were not reported before. Namely, we are starting to see a clear solvent network, which allows us to see water molecules decorating the interface between NSP1 and the ribosome. And they actually reveal water-mediated contacts to both ribosomal proteins and ribosomal RNA. And even a water molecule is visible here that is bridging those 2 NSP1 helices. We can also start seeing additional features such as PTM at the bottom of the mRNA channel, where the NSP1 binds, as well as some metals, none of which were present in the deposited PDBs. Perhaps the most interesting feature now visible at high resolution is this water molecule position here at the hotspot of the interaction between the NSP1 and the ribosome. This is the well-known KH motif, and we actually start seeing water molecules around it. Again, this is still at early stage, but we hope that with improving resolution, we will see even more structural details. Just at this point, I want to go back to the first question we always get about cryo-arm is if 1.8 angstrom BetaGal is the limit. Well, I'm glad to inform you that we actually obtained 1.6 angstrom BetaGal map. Now, this jump from 1.8 to 1.6 is pretty significant when you get to this high resolution range. And needless to say, the map shows even greater and better structural differences, second hole in tryptophans, nicely resolved other features. And, you know, at this point, actually, we start seeing, although BetaGal is considered a benchmarking protein, when you get to this high of a resolution, you actually start seeing some interesting chemistry. And at this point, I'm not going to discuss that because we still want to make sure that we interpret the data correctly. But it's actually pretty interesting. It was nice to get BetaGal to 1.6 angstrom resolution, but the question was, can we do the same with the ribosome sample? Can we push the resolution there? And we decided to take a shot at the rabbit ribosome, ribosome purified from rabbit reticulocytes. And we actually managed to obtain a map at 1.75 angstrom resolution. Shown here is the local resolution plot that shows that actually in the core of the particle of large ribosomal subunit, the resolution extends past 1.6 angstroms. This represents the highest resolution ever obtained for ribosome from any species using any structural biology method. At this high resolution, you can see usual features, as I mentioned so many times before, but just everything is so crisper and it's easier to correctly identify the solvent particle and to interpret extra densities as post-transcriptional modifications and post-translation modifications. After seeing these many structural details and after we saw this great push in resolution for rabbit ribosome, we went back to human ribosome and decided to go one step further and to add the only FDA-approved drug targeting human ribosome directly, and that's AlmostToxin, also known as homoharingtonin. This drug is used for treatment of chronic myeloid leukemia. The structure of AlmostToxin bound to ribosome was first done using crystallography in Stites lab, but the ribosome used was archaeal. And it was also done in complex with human ribosome using Cryo by Bruno Plachholz, and it was resolved at 2.9 angstrom resolution. The overall resolution we got after data collection on the Cryo 200 microscope was at 1.8 angstrom resolution. And needless to say, the density looks really good, showing all the high resolution features I already showed on previous slides. But here the focus is on the drug. And even though the drug was added in access, we can still see substoichiometric occupancy. The ligand density is nicely resolved. We can start seeing holes in rings. However, the tail of the drug is a little bit washed off. When it comes to the binding site, the drug is obviously bound at the same spot as previously reported. However, we are seeing certain differences. Potassium ion, for example, is differently positioned than in Stites map, and there are several residue flips. So even though mechanistically it looks the same, chemistry and crucial interactions are actually different. Yeah, we are even seeing the different solvent positions in important parts of where the drug binds. Even though where the drug binds, that's not the best part of the map, the density for the drug is, I would say, really shows really nicely all the features. And again, the local resolution plot shows that the core of the large ribosomal subunit it actually extends past 1.6 angstrom resolution. And here are just some high resolution features we are seeing in this map. Again, this is the methylated histidine that I already discussed, and it is obvious that this histidine 245 is again methylated. Density just shows this methylation quite clearly. And there are some other high resolution features that we are seeing in this map. And I would like to close by thanking my team, Alan Merck, who spearheaded the tech dev on our Cryo 200, Joseph Darling, Reichel Wieshammer, and other friends and colleagues at FNL. I would also like to thank a lot to Takuma Fukumura, who was actually instrumental in writing all the serial YAM scripts we use for data collection. and who was our sensei in learning how to use this microscope. And I would also like to thank Shriram Subramaniam and Xing Zhu from UBC for the help. They helped us with 1.8 Angstrom BetaGal Map. And with that, I would like to take your questions.